High school biology textbooks present cellular respiration as a straightforward transaction. Glucose goes in, while ATP and carbon dioxide come out. This treats the metabolic pathway like a mechanical furnace designed solely to break down fuel. This breakdown-only model overlooks the anabolic side of the cellular ledger. A cell must extract energy by breaking carbon bonds, while simultaneously preserving those carbon backbones to build new physical structures. The pathway manages this by routing both catabolic and anabolic traffic simultaneously, operating as an amphibolic network. The citric acid cycle acts as the cell's transit hub for a continuously calculating carbon economy. Every structural component of a cell requires a specific carbon skeleton. The ribose sugars in RNA require a different structural arrangement than the hydrocarbon tails of lipids. The raw materials for these diverse structures originate from the same central metabolic network. To understand how life sustains itself, we must map the stoichiometry and flux routing of these carbon intermediates. Carbon routing begins in the cytosol with the arrival of a 6-carbon glucose molecule. This diagram shows the initiation of the pathway. Hexokinase adds a phosphate group to the 6th carbon, which traps the glucose inside the cell. Aldolase then splits the 6-carbon ring into 2 3-carbon molecules. This cytosolic phase yields a net ledger of 2 pyruvate molecules, 2 ATP, and 2 NADH. Pyruvate follows different fates depending on oxygen availability. Under anaerobic conditions, it is reduced to lactate to regenerate NAD+. Under aerobic conditions, it is transported to the mitochondria. Glycolysis functions as a preparatory phase, securing minimal energy while staging carbon backbones for the primary mitochondrial engine. We now follow the journey of the pyruvate molecule as it crosses through the dense inner mitochondrial membrane and descends deep into the matrix. During oxidative decarboxylation, pyruvate loses a carbon as CO2 to become the 2-carbon acetyl-CoA. Acetyl-CoA merges with the 4-carbon acceptor oxaloacetate, forming the 6-carbon citrate. Moving through intermediate stages, it returns to a 4-carbon structure by releasing CO2. Simultaneously, this cycle harvests high-energy electrons, transferring them to NAD+ and FAD to power the later stages of respiration. The cycle's catabolic role is essentially an electron harvesting mechanism, dismantling the acetyl group to power oxidative phosphorylation. Cataplerosis is the process of draining intermediates out of the cycle to support anabolic construction. In this map, citrate is diverted out of the mitochondria to provide the building blocks for fatty acids and cholesterol. Alpha-ketoglutarate is extracted to synthesize glutamate, which provides the carbon skeleton for the purine bases of DNA and RNA. If these intermediates are exported too rapidly, the oxaloacetate acceptor cannot be regenerated and the energy cycle halts. Anaplerosis provides the refilling reactions. Pyruvate carboxylase synthesizes oxaloacetate from pyruvate to restock the cycle. Cellular survival depends on balancing this catabolic energy extraction with the cataplerotic demands of growth. An error in this network corrupts the cell's identity because the metabolic pathway provides the physical building blocks for all cellular structures. This is visible in the neomorphic mutation of the enzyme isocitrate dehydrogenase, or IDH. Under normal conditions, IDH oxidizes isocitrate to produce alpha-ketoglutarate. But the mutated enzyme abnormally consumes NADPH and generates an aberrant molecule called 2-hydroxyglutarate. 2-hydroxyglutarate is an oncometabolite, a metabolic product that actively drives the formation of tumors. As this toxic oncometabolite rapidly accumulates within the mitochondrial matrix, it physically leaves the mitochondria and forcefully enters the cell nucleus. In the nucleus, it acts as a competitive inhibitor against TET enzymes. TET enzymes are responsible for hydroxylating methyl groups on DNA to facilitate demethylation. When TET is inhibited, the DNA undergoes rapid hypermethylation. This structural hypermethylation physically locks the cell in a primitive, undifferentiated state, rapidly promoting aggressive and uncontrolled cancer tumor growth. The sequence of DNA encodes the enzymes of metabolism, and the metabolic network generates the carbon intermediates that regulate the expression of that DNA. These two systems form a feedback loop of form